Greetings, students. Mr. Sanger, live from my living room this time instead of the storeroom as I prepare a screencast getting us ready for a lab. This lab is uniquely suited for a distance learning environment because it's pretty much all paper pencil. So let's get going and figure out what this is all about. So it's called Factors Affecting Mitosis. So the two main objectives. One, you're going to analyze the effect of two variables on the rate of mitosis. And those two variables are, one, the exposure of cells to chemicals called lectins, and the second variable is cancer. And then you're going to perform a statistical test called a chi-square test to analyze experimental results. And so I recognize that is a new thing, so much of this screencast is going to be devoted to talking about what is this chi-square business. So first, some background. Lectins. Lectins are a protein produced by fungal pathogens, and they're suspected of affecting the growth of soybean plants by inducing mitosis. And so to investigate this, you're going to look at the rate of mitosis in cells that have been treated with lectin and the rate of mitosis in cells that have not been treated with lectin. So how was this data acquired? Well, what happened is someone would have to get this data, would need to actually look at cells in the midst of mitosis. And so what they do is they set up an onion, regular old onion that you'd buy at the grocery store, and they can grow it in some solution. This is called hydroponics, with nutrients and whatnot. And then the roots are going to grow into this nutrient-rich solution. So if the roots are growing, by golly, mitosis must be taking place. And so what they do is they take that little tip of root, they cut it off with a scalpel, and then they slice it into really thin sections, and they stain it with some dyes and chemicals that kind of stain the nucleus so that you can see the DNA. And the microscope slide would look something like this at fairly low power. And so all these little black circles, that's actually the DNA, that's the nucleus of the cell. Now you may look at that and think, what am I can't see. Well, let's zoom in closer. If we zoom in closer, this is what it looks like. And you can start to see, oh, that's like an individual cell. These little boxes are the cell walls. And then that is a cell in a particular phase of mitosis. And over here we have a guide of what the phases look like. So we start in various phases of interphase here. In interphase, the cells are, the nucleus, just looks like kind of a solid black circle. So this is a cell in interphase, this is a cell in interphase. And it looks like that because the DNA is in this uncoiled state called chromatin, which is basically like taking your rope and just throwing it in a big pile. So, so it just kind of looks like a black circle, but really it's this, rope-like DNA filling out that entire nucleus. By about cell 3, 4, 5, the chromosomes, or the DNA is starting to condense into these chromosomes. So you can start to see these kind of distinct lines forming, and those are the rope being all coiled up into a nice tight chromosome. And so I'm not sure, like maybe this is kind of a prophase sort of a thing. This one's fuzzy, but I think that's a prophase. Metaphase, the chromosomes are all lined up down the middle of the cell. And then in anaphase, the chromosomes start to move apart to opposite ends of the cell. So this one that I started with is a beautiful anaphase. This one looks like a metaphase. And then eventually the cell wall starts to get built down the middle of these two, and we have a telophase. And so basically what you would do for this is you would look in a microscope and you'd basically go down the line, like interphase, fuzzy, can't tell, prophase, interphase, and you just kind of make a little tally of how many cells are in each phase. If you've ever seen the movie Twilight, apparently I'm told there's a scene where they're actually doing this lab while there's some like teen I don't know, there's like flirtatious activity going on during this lab, I don't know. Anyway, so that's how the data was collected, and in the lab itself you will see numbers of how many cells were in each phase of mitosis for cells treated with lectin and cells not treated with lectin. So now the next question will be, how do we tell if there's actually a difference between the cells treated with lectin and the ones that aren't? And so to determine if the differences are statistically significant, we need a new statistical test. And that test is the chi-square test. This is a statistical test that compares expected results to observed results to see if the difference between them is statistically significant. In other words, is any variation that we see due to random chance, or is that variation due to a variable we manipulated? The chi-square test will tell us that. How does it work? Well, let's start by flipping a coin, because that's something that most of us are familiar with. Even as a teacher at my low salary, I have coins available that I can flip. So imagine that you flip a coin 20 times. What are your expected results? I'm guessing you would expect to get a perfect 10 heads, 10 tails, right? Because there's a 50-50 chance, so half the time you get heads, half the time you get tails. So 10 and 10 are the expected results. Well, anyone who's ever flipped a coin knows that it's probably not likely that you're going to get exactly 10 heads and 10 tails. So let's show some actual data. And the question is the difference in the data shown, is the difference significant? Is it statistically significant? So, for example, let's suppose we flipped a coin and we got 18 heads and 2 tails. I think most of us would agree that's a little weird. Like, something seems like maybe it's a weighted coin, or there's something going on with that, because that's a really dramatic difference. And then you do it again with another coin and you get 12 heads and 8 tails. And I think most of us would think, well, 12 and 8, that's pretty close. That's probably, just random chance causing that variation. And so the question really is, where's the line? Like, what if it's 13 and 7 or 14 and 6? Like, where is it between 12 and 8 that we say is pretty much random chance, and 18 and 2, which we say, it's definitely not random chance, something's going on there? At what point can we tell? That's where the chi-square test comes in. So, how do we do it? This is the formula for chi-square. I want to point out that this formula is on your equations and formula sheet for AP Biology. You got this in the early days of class. It's also available on Schoology. And lo and behold, there is the chi-square test. And then the chi-square table, which we're going to look at in a moment, is down there. So you have this, certainly on any test for AP Bio, you want to have that handy. So there's the equation, and it looks intimidating and freaky. The x with the square, this is the Greek symbol chi, and then it's squared like that. This means the sum of, and then O is observed values, and E is expected values. Okay, so how do we break this down? So step one in using the chi-square test, you're going to state what is called the null hypothesis. The null hypothesis always means there is no significant difference between our observed and expected value. In other words, our variable has no impact at all. So if we flip a coin 20 times, how many heads and how many tails would the null hypothesis predict? Did you say 10 and 10? Because it's 10 and 10. 10 and 10 means there's no statistically significant difference between observed and expected. It was 10 and 10, that was observed, and we expected 10 and 10, right? So what we need to do, what if we have 12 and 8, for example? Well, step two is we need to do some math to actually calculate the chi-square value. So again, you have this formula on your formulas and equation sheet. And so all you need to do is plug those numbers in. So let's do it for this particular set of data, this 12 and 8. If we want to calculate chi-squared for that value, we would use these numbers right here. So let's switch to the dot cam. Look at this fancy stuff, and oh my gosh, look at that. The formula is all ready for us. So if we have 12 heads, 8 tails, since we're taking the sum, we would start to plug in numbers. Our first observed number is 12, right? We observed 12 heads. Our expected number is 10, because if there's 20 flips, we expect 10 heads, 10 tails. So I go 12 minus 10 squared over expected, which is 10. But then it's the sum of, because I have this other value here. So then I'm going to add the next one. So my next observed value is 8, minus expected is 10, over expected is 10. And so what you do is you calculate the actual value. So 2 squared over 4, it's like 410 over 10 plus 4 over 10, and that's 8/10, which, gosh, math fractions. How did I do? 0.8. I think that's the number I got. And so because I never trust myself, I always put the answer on my slide, and oh my gosh, there it is. So we calculated the chi-square value as 0.8. Okay, so what do we do with that? Well, step 3 is we now decide if we should reject or fail to reject our null hypothesis. And we do that by comparing the chi-square value to this critical value in the table below. So to do that, you look, and it's asking for two things, a p-value and a degrees of freedom, and then there's all these weird numbers. What the heck is going on? Well, degrees of freedom is always n minus 1, where n is the number of outcomes. So when you flip a coin, there's two outcomes. Either it's going to be heads or it's going to be tails. So n minus 1 would be 1. So our degrees of freedom is 1 in this case. Later we're going to do these things called Punnett squares, where there might be 4 outcomes. So then n minus 1 would be 3. The p-value, we're always going to use 0.05 in this class. And what that means is we're looking for 95% confidence in our answer. In other words, if we did this experiment 100 times, 95 out of 100 times, it would agree with what we decide. Sometimes people want more precise p-values. For example, if you're designing a medication, you want better than 95%. You want it to work, 99.9999% of the time. The M&M factory actually has a formula to make sure that there's a proper ratio of each color of M&M so that it looks visually appealing to the consumer. But for that, it doesn't matter that much, so their p-value might be 80% because maybe it's too expensive to have it exactly in the right color ratio. So the p-value kind of varies, but in this class, 95%. So p-value of 0.05, 1 degree of freedom, our critical value is 3.84. Here's the rule. If chi-square is greater than our critical value, we reject our null hypothesis. If it's less than our critical value, we fail to reject our null hypothesis. In other words, if chi-square is bigger than this, it's a big deal. Something significant is going on. If it's less than this, it's not a big deal. There's nothing significant going on, so we fail to reject. And if we fail to reject the null hypothesis, it means, well, the null hypothesis is probably accurate, which means there's really no difference going on. So if you look at our numbers, did we reject or fail to reject? Well, our value was 0.8. The critical value was 3.84. So chi-square is less than our critical value. We fail to reject our null hypothesis, which means we're basically saying there's really no statistically significant difference between a coin flip of 12 and 8 and a coin flip of 10 and 10. And that, I think, meets your expectations, right? Because 12 and 8 is pretty close. So let's go to this 18 and 2 one. I will let you go ahead and plug numbers in and see if you can calculate the chi-square value, use the degrees of freedom to determine if you should accept or reject your null hypothesis, and then maybe pause the video to do that. And then, you ready for the answer? 12.8. So 12.8 is going to be larger than the critical value of 3.84. If it's a bigger number, that's a big deal. Something's going on. That means that we reject the null hypothesis, which says nothing's really going on, and we say, okay, there's something going on here. This coin, there's something going on with that coin. These numbers are not 10 and 10. Something significant is happening. So if this is all very odd and confusing, you might want to pull these slides up on Schoology and click that link. Here's another person's take on that chi-square test. Just want to remind you of the science practices that we're engaging with: statistical tests, data analysis, then yeah. So at this point, you're ready for the lab. You're going to be analyzing data, doing some chi-square, and drawing some conclusions. Good luck.